Good afternoon. This is Mr. Siwale. I want to talk about the endocrine pancreas, specifically with focus on the action of insulin. The endocrine pancreas with specific concentration on insulin. Okay, first and foremost, this diagram shows the insulin producing gland, which is called the endocrine pancreas. Okay. This pancreas is a triangular gland which has both the exocrine and endocrine cells, which is located behind the stomach. Okay, so it is at a strategic location. So histologically, if you remember from your histology, the acina cells produces an enzyme-rich juice, which is used for digestion, which is called the exocrine product or the pancreatic juice. However, the pancreatic islets, they produce hormones involved in regulating fuel storage and the use. And basically, the hormone of importance here we are talking about is insulin. Okay, so that is our endocrine pancreas. When you look here, this is just a histological aspect of the islets of Langerhans. So the insulin-producing cells are called pancreatic islets, okay, or islets of Langerhans. Remember that not only insulin is produced from these islets of Langerhans, okay. There are even other cells. There are even other hormones which are produced by this pancreas. We have glucagon, polypeptide Y. We have insulin itself, and there's this fourth hormone which is called there is insulin, somatostatin. Yes, it's called somatostatin. So in terms of percentage, these hormone producing glands of the pancreas, they are just about 2%. The rest, the 98%, they are what we call the cells that produce the digestive juice. Okay. They are the cells that produces the digestive juice. So this is the structure of insulin. If you remember from the introduction, I said insulin is just a peptide hormone. Okay, it is a peptide hormone. So to which class does it belong to? This belongs to the class that is made up of amino acids. Okay, so it's a peptide hormone. So from here you can see that insulin has it has A and B peptide chains. Okay, it has the A, and this is the B peptide chains, and this is the A peptide chains, which are connected by disulfide bonds. Okay, these are disulfide bonds. But however, the precursors of this insulin is called the proinsulin. Okay, before the insulin is synthesized, there is a precursor which is inactive. It's called the proinsulin. Okay, sorry about that. It is called the proinsulin. Now, when you look at synthesis of insulin, the synthesis of insulin by the pancreatic beta cells, it follows the first step. It happens in the ribosome. This is where preproinsulin. Okay, it's where preproinsulin is synthesized. Preproinsulin is the precursor of insulin, and then the second, it happens in the endoplasmic reticulum, where the preproinsulin is changed to a proinsulin by enzyme which are called microsomal enzymes. And then from there, it moves into the Golgi apparatus. The proinsulin enters the Golgi apparatus, where it is packaged into the secretory granules. And then the fourth, the secretory vesicles is called secretory vesicles. Inside the secretory granules, proinsulin is cleaved into insulin, which is made up of 51 amino acids, and the C peptide, So it's important to note that the insulin and the C-peptide are secreted in equimolar amount. So if you can measure C-peptides in a patient, it's the same as knowing the concentration or the amount of insulin being secreted. That's why we mean that they are secreted in equimolar amount. So being a protein, it follows the general synthesis of proteins. Now look at this. What are the main substances that regulate insulin secretion? What are the main substances that regulate insulin secretion? So the main potent stimulant of insulin secretion is glucose. Okay. And then the major inhibitor of insulin secretion is somatostatin. Actually, somatostatin, this hormone, inhibits many other hormones, not only insulin. So one of the functions of the somatostatin is inhibitory function. Okay, so the major potent stimulant of insulin secretion is glucose. This glucose is found in the diet. That's why every day we are always eating glucose-containing foods. Okay, the reason is because we want insulin secretion, and insulin helps in absorption of glucose into cells. The others, we have mannose, amino acids such as leucine, arginine. Okay, we have intestinal hormones such as GIP. We have GLP-1, gastrin, secretin, CCK. Okay, these are inhibitory. They can stimulate synthesis of insulin. We have beta keto acid, acetylcholine from the parasympathetic secretions. We also have glucagon, stimulates insulin secretion. For example, when the glucagon levels reduces, okay, insulin will be stimulated. We also have drugs such as sulfonylureas, okay, such as Downhill. They also stimulate insulin secretion. So these are used in treatment of diabetes mellitus. Okay, it's just important to know this. But we also have factors. Let's start from down here. The alloxan. Alloxan is known to destroy the pancreatic beta cell, and it is very much used in research to do with diabetes mellitus, because alloxan destroys the pancreatic beta cells, so there is no insulin production. So when there is no insulin production, it leads to a condition called diabetes. So this is used in research. Okay, these we will not discuss, but it's just important to know that these factors, they can stimulate insulin secretion. So by what mechanism is insulin secreted? We'll just run through this. The first is glucose. It enters the cells through the glucose transporters. And inside the cell, there's this, which we call glucokinase, converting glucose to glucose 6-phosphate. And then we've ignored certain reactions. Glucose 6-phosphate can be converted to fructose 6-phosphate. Fructose 6-phosphate can be converted to fructose 1,6-phosphate. Fructose 1,6-phosphate can be converted fructose 1,6-phosphate. If you remember your glycolysis, it's cleaved to form diacylglycerols, okay, and dihydroxyacetone. Okay, just to revise the biochemistry. And then, but what we're interested is in the formation of ATP. Okay, so what does ATP do? It blocks the potassium channel, specifically the SUR1. This means sulfonylureas subunit of the potassium channels. So potassium doesn't leave the cell becomes depolarized. When the cell becomes depolarized, calcium enters. So calcium enters, will will combine with the insulin containing vesicles to trigger exocytosis. Okay, so this diagram just shows the mechanism by which insulin is secreted from pancreatic beta cells. So if you remember, people with diabetes, they have insufficient insulin secretion. So any drug that can result into inhibition of the potassium channels is a very good drug for treatment of diabetes. Some of them are glibenclamide. Glibenclamide, if you look at the mechanism of action, it blocks the potassium channels, these. Okay, so there's potassium doesn't leave the cell, resulting into depolarization of the pancreatic beta cells, and then resulting into entry of calcium into the cell and triggering exocytosis. Okay, and then we have this insulin receptor. Okay, this is the one we are calling the tyrosine. So the insulin receptor is known as a tetramer. Okay, tetramer. Okay, tetramer like 4, which is because, why is it called a tetramer? Is composed of 2 alpha subunits, these ones, and then it is composed of 2 beta units. So together they make 4. That's why it's called a tetramer. Okay, the alpha subunit, as you can see, they lie in the extracellular domain. Okay, and the beta subunit of the insulin receptor, they span the cell membrane like integral protein. And at the end of the beta subunit, there is this enzyme called the tyrosine kinase. Okay, there's a enzyme called the tyrosine kinase. You may wonder, what's the function of the tyrosine kinase? This tyrosine kinase, it belongs to the classes of cell receptors. Okay, the cytosolic domain, the one which lies in the cytosol, activates the cytosolic kinases. Okay, so that translocates to the nucleus and activates nuclear transcription factors by what we call phosphorylation. So if you hear the word kinase, it means there is a phosphorylation taking place, addition of a phosphate group, and we know phosphates are associated with energy. Okay, so tyrosine kinase is an enzyme that activates nuclear transcription factors by phosphorylation. So when insulin binds to this point there, okay, the tyrosine kinase becomes phosphorylated. It becomes phosphorylated. The mechanism we call autophosphorylation. Okay. So when it becomes phosphorylated, that's when we are going to see physiological actions. For example, production of glucose transporters. Okay, if it is in the liver, insulin will cause storage of glucose, that is conversion of glucose to glycogen, and all those biochemical reactions will take place. Okay, if it is in the adipose cells, insulin will cause adipogenesis, okay, whereby glucose is converted to adipose, okay, and it's stored as fat. So this is our insulin receptor. So this is a proposed mechanism of action of insulin. Okay, this is not very important for now. Okay, it's not important, it's just a proposed mechanism of insulin action. But I'll just run to where are the effects. We are interested in these effects. This is what is important for now. Okay, these are different cascades of reactions, but we are interested in these effects. Okay. So what is the effects of insulin? It increases insertion of glucose transporters in the cell membrane, as you can see here. The other effect, it increases lipogenesis. Okay. Lipogenesis is the production of lipids. The other effect of insulin, it decreases gluconeogenesis, decreases glycogenolysis, that is the breakdown of glycogen. It increases adipogenesis, and it can increase or decrease adipokines depending on the situation. So I would want you to know these actions of insulin. These are what we call the metabolic actions of insulin. Okay, it has effects on the carbohydrates, it has effects on the lipids and proteins. So insulin affects the metabolism of carbohydrates and proteins. This is what I want you to understand. It affects the metabolism of glucose or carbohydrates and lipids. Okay. So in this table form, we have summarized the action of insulin. We can just run through them. What's the action of insulin? It increases glucose uptake into the cells. Okay. It increases glycogen formation. It decreases glycogenolysis. It decreases gluconeogenesis. It increases protein synthesis. Okay. So it has an anabolic effect. For example, the glucose transporters, those are proteins, and they are needed whenever insulin binds to the insulin receptors. And then it increases fat deposition. That's why too much carbohydrates results into increase of fats. Okay, because high levels of carbohydrates means there's high levels of glucose in the blood, which stimulate insulin secretion, and insulin is storage hormone. It decreases lipolysis. Instead of promoting breaking down of lipids, insulin decreases that. Okay, it also increases potassium uptake into cells. So insulin causes, is it hypokalemia or hyperkalemia? Insulin causes hypokalemia. When there is increase in potassium uptake into cells, it means there will be decreased levels of what? Of potassium. Okay, this is physiology. Make sure that you understand this. Okay. The only way to understand this is just to know. There's no formula about it. You just have to know about it. Thank you very much. Okay, these are just diagrams showing you the effect of insulin, lipogenesis, and lipolysis. Okay, lipogenesis and lipolysis. So what can we see from these diagrams? So like, let's see. Insulin promotes lipogenesis. So under lipogenic conditions, insulin binds to the insulin receptors, okay, located on the surface of the adipocytes, and transfers glucose transporters to the plasma membrane. These glucose transporters to the plasma membrane. So these adipocytes will take glucose into the cell, and when glucose is entered into the cell, it is converted to glycerol 3-phosphate or glycerol 3P. Okay. And you know glycerol 3-phosphate is needed for production of triglycerides, which are stored in adipocytes. Okay, so that's how the mechanism by which insulin causes lipogenesis. Okay, it increases uptake of glucose into the cell, And once the glucose is inside the cell, the glucose is converted to glycerol 3-phosphate. So it activates the enzymes that converts glycerol 3-phosphate. Some of these enzymes that insulin activates are things like lipoprotein lipase, okay, located on the cell surface of the vascular endothelium. So once this lipoprotein lipase is activated, it helps absorption of chylomicrons, okay, these chylomicrons, and very low density lipoproteins. Okay, so this is just the opposite, lipogenesis, lipolysis. One of the hormone that contributes to lipolysis, it's whenever there is low levels of insulin, like glucagon. So under lipolytic condition, we can have catecholamines. We know these are released by the sympathetic nerves. Catecholamines such as epinephrine. They will stimulate adrenergic receptors located in the surface of the adipocytes. And the activation of these, when it binds to this receptor, it causes activation of hormone-sensitive lipase, this one, which is the key enzyme in breaking down triglycerides to free fatty acids and glycerol. Okay, so what's the effect of catecholamines? It activates this enzyme, the hormone-sensitive lipase, and also aquaporins P7. They are synthesized. Okay, so it is just important to understand the mechanism of action of this insulin. So we can summarize the action of insulin in this table. Whenever the insulin levels go up, the beta cells will be stimulated. They'll become depolarized. This will result into exocytosis of insulin from the beta cell. So insulin will act on the liver, it will act on muscle and adipose and other cells. On the liver, it changes the biochemistry or metabolism of this important substrate. Okay, so it decreases glycolysis. On the liver, whenever the plasma levels increases, it increases glycolysis. It increases glycogenesis, that is breaking down of what is glycogenesis? Breaking down of glycogen into glucose. That is glycogenolysis. Now this is glycogenesis. So insulin increases glycogenesis, that is synthesis of glycogen. It increases lipogenesis, synthesis of lipids. It increases glycolysis, okay, which is synthesis of ATP. That's the action of insulin on the liver. On the muscle, it increases glucose transport. So the effect of this will be low glucose levels. And the low glucose levels will have a negative effect on the pancreatic beta cell. Okay, which is what we call a negative feedback mechanism. Now this, we cannot end without talking about glucagon. Okay. Now, glucagon has the opposite effect of insulin. We need to know that. We need to remember that. It has the opposite effect of insulin. So in other ways, it opposes the hepatic effects of insulin. Okay? So it's very easy to remember this. So pancreatic glucagon opposes the hepatic effects of insulin. Okay? and whereas the GLP acts as incretins, which are gut-derived peptides that enhances glucose-stimulated insulin secretion. So the secretory half-life of glucagon is 3 to 6 minutes. You may ask what is GLP? It's glucagon-like peptides. Glucagon-like polypeptides, if you want to call it that way. Okay, so glucagon has the opposite effects. The mechanism by which glucagon increases glucose output is just the opposite of those which we described with insulin. But we need to understand the enzymes that glucagon does activate. Glucagon activates gluconeogenesis, which is the reverse of glycolysis. Not really reverse, but is most of the steps of glycolysis are reversed. So glucagon reverses some of the steps in glycolysis. Okay, and glucagon also converts. First of all, this is activates. This line shows activation, and then the dashed lines indicates inhibition. So glucagon inhibits the formation of fructose 2,6 biphosphate, which is needed in glycolysis to proceed all the way until pyruvate. So it inhibits the synthesis of this. Okay. So, however, glucagon acts on different glucagon receptors located on the same hepatic cells to activate phospholipase C, and the resulting increase in cytoplasmic calcium also stimulate glycogenolysis. Okay, this is just information which you can read on our own. Now, what factors stimulate or inhibits glucagon secretion? This is also important. Exercise stimulates glucagon secretion. Cortisol, stress hormone, stimulates glucagon secretion. Amino acids, particularly gluconeogenic amino acids like alanine, serine, glycine, cysteine, and threonine, they stimulate glucagon secretion. Okay. So in other words, factors that inhibit insulin secretion, you find that those factors, they can stimulate glucagon secretion. And factors that stimulate insulin secretion, those factors can inhibit glucagon secretion. Okay. Now, what about GABA? GABA inhibits glucagon secretion. The release of GABA coincides with the release of insulin secretion produced by hyperglycemia. So whenever insulin is secreted, there is also GABA which is secreted. The GABA is released and acts on the A cells, the glucagon producing cell, to inhibit glucagon secretion by activating the GABA-A receptors. So these GABA-A receptors are chloride channels, and you know whenever chloride enters into the cell becomes repolarized, and it's very difficult for exocytosis to take place, and the resulting chloride influx hyperpolarizes the A cells. Okay, so you may be wondering what happens to glucagon when insulin is being secreted. It is inhibited via GABA. Okay, we cannot also end this without talking about diabetes mellitus, although some of these things will encounter them in pathology, but it's important to say that diabetes mellitus is a commonly known sugar disease. It is characterized by long-term recurring high blood sugar levels, which is called hyperglycemia. Therefore, it can be defined as a disease characterized by chronic hyperglycemia state due to defects of insulin secretion, insulin action, or both. We know that insulin helps in absorption of glucose, so whenever insulin is deficient, the glucose levels in the blood increase. So the causes of diabetes, especially type 1 diabetes, is degeneration of the pancreatic beta cells in the islets of Langerhans, destruction of the pancreatic beta cell by viral infection, congenital disorders of the pancreatic beta cells, destruction of pancreatic beta cells due to autoimmune disease, where the immune system recognizes the beta cells as foreign and starts to destroy them. So this occurs in children. That's why it's called type 1 diabetes or juvenile onset diabetes. Now there's type 2 diabetes. This one is associated with lifestyle, whereby as we grow, as we are in exploring life, we eat all sorts of food. We do not exercise frequently. We don't care about what we eat, whether we eat healthy meal or not. So in this type of diabetes, the structure and function of beta cells and blood levels of insulin are normal, but insulin receptors may be less, okay? They are down-regulated. They may also be absent or abnormal, resulting in insulin resistance. Okay, so you find that the insulin receptor, that tyrosine kinase we saw, may become down-regulated, may be absent or normal. What can cause down-regulation of this? It can be genetic disorders. Okay, it can also be lifestyle changes such as bad eating habits and physical inactivity leading to obesity. So obesity is a risk factor for type 2 diabetes, and stress. Okay, this is a very interesting subject because it mainly talks about metabolism. It's a disease of metabolism, glucose metabolism and lipids metabolism. Okay, we'll still be exploring these as we go on. So what goes on wrong at physiological level, from the physiological point of view? In diabetes, the fasting blood glucose levels are greater than 126 milligrams per deciliter. Okay, but we know that this shouldn't be the case in a normal healthy person. In a normal, it should be 100. It should be less than 100 milligrams per deciliter, the fasting blood glucose. Again, if a test is carried called the 2-hour oral glucose tolerance test, a person with diabetes, the result will be greater than 200 milligrams per deciliter or 11.1 millimole per liter, which is, these are SI units. The millimoles, they are SI units. In pre-diabetes, about between 140 to 100, but in a normal person, it should be less than 140 milligrams per deciliter, the 2-hour oral glucose tolerance test. There's this another diagnostic criteria called the glycated hemoglobin. Okay, you know the sugars can bind to hemoglobin. It's a protein, so they form what you call glycated hemoglobin. So this can be measured. Okay, in a diabetic person, if you find that 6.5% of the hemoglobin is glycated to glucose, it means that's diabetes. In pre-diabetes, that is before onset of diabetes, is between 5.7 to 6.4%. But in a normal person, yes, there's some glycation of hemoglobin, but it's between 4% to 5.6%. Okay. So it's important to compare the normal to the abnormal. So as physiology class, we are interested in understanding the normal before we can appreciate the pathology. Okay, so when a person has diabetes, usually these are the symptoms. Okay, due to insulin insufficiency, insulin resistance, and hyperglycemia, diabetes cause the following: hyperglycemia, glucagon excess, ketoacidosis, diabetic coma, and there's what we call classical triads called polyuria, polydipsia, polyphagia, and can lead to hypotension. Okay, so it can also lead to the formation of ketone bodies, which we call ketoacidosis. So the acids in the body increases the acetones, they increases. Okay, and those other ketone bodies, the beta hydroxybutyrate and the acetoacetic acids, they increase. Because in this case, glucose is not entering the cells, but lipids become the alternative source of energy. So the lipids are broken down in beta oxidation in the mitochondria, resulting into production of the beta hydroxybutyrate and acetoacetic acids. Okay, so this is where we end. At least we know insulin, the action of insulin. It helps us, or it helps the body to absorb glucose. Okay, it also helps to store glucose, which we call lipogenesis. Okay, so any defects in insulin results into diabetes mellitus, which is becoming a very common disease. Okay, thank you very much.